Hello everybody, my name is Tom Helikar and I'm here today to share with you kind of my journey through my professional and scientific life and talk to you about computation and bioinformatics in life and health sciences. First of all, I would like to thank the organizers for having me here today and giving me the opportunity to share my journey with you. So I am currently a assistant associate professor in the Department of Biochemistry at the University of Nebraska in Lincoln. And to give you to start with my journey and sharing my journey with you. So I was probably, as you can tell by my accent, I wasn't born in the U.S. I was born in the Czech Republic, in particular in this city called Brno. It's Czech Republic is right in the middle of Europe. And I first came to the US when I was 18, when I was an exchange student in high school. I spent 1 year in high school in Omaha. And after that 1 year, I went back home and I took my differential exams in the high school I was in the Czech Republic. And right after that, I started my undergraduate career at the University of Information Technology in Brno in the Czech Republic. This was in 2001. And I spent about a year and a half to 2 years as an undergraduate student, majoring in computer science and computer engineering. And then I went back to Omaha. And at that time, my plan was to go back to Omaha just for a semester. I got accepted in a community college and the goal was to improve my English in the field of computer science. But then I met my wife, and 20 years later, I'm still in Nebraska. But also during that time, I explored the University of Nebraska in Omaha, and in particular the computer science program. And so I had a chance to transfer to that program, and uh, it was during one of my mathematics classes. In particular, it was Math 2030, Discrete Mathematics, that I met Jim Rogers. He's now actually a chair of the Department of Mathematics at UNO. And he was teaching this class, and at that time, I knew I was uh, going to do computer science, and thinking that I would uh, end my graduation and start my career by working for a company writing software. Uh, but then when I met Jim, he just at that time he got a big NIH grant to study biological systems, and part of that grant was to develop a mathematical model of signaling networks of pathways that happen inside the cells that help cells sense the information. And so I accepted that position, or I applied for a undergraduate research assistant position, and he took me, and I told him I don't know anything about biology or biochemistry. I was a computer science student, and in fact, biology was not on the list of my favorite classes. And so, there was a big, steep learning curve for me at that time. And so I started working on that. And that's really when I started realizing the need for computational biology, bioinformatics, and computer science in biology. And so let's talk about that. Why do we need computer models and computation in biology? Uh, it turns out that biological processes and organisms are extremely complex. Over the last several decades, the scientific and health sciences community have been extremely good and productive at generating tons and tons of data. This chart here just shows you the sequences, DNA sequences, that have been deposited year over year in public databases. Okay, and you can see this exponential growth of these sequences. But it's not just at the level of DNA and genes. This complexity and this amounts of data we are generating at all levels of the biological organization, whether it's genes, proteins, cells, organs, organism, populations. We're generating lots and lots of data. And this is in the life sciences. If we look at also the health sciences, at the physiological level, we're talking about wearable devices, um, devices that can measure your blood pressure or glucose or calories you're burning and things like that. We're talking about these devices to also to continue to grow, and so we will continue to collect data on a day-to-day basis. Even non-scientists, just general population wearing these devices will be collecting and generating enormous amounts of data in addition to the scientific community that generates data at the gene and protein and cellular level. And so life sciences and health sciences have really become information sciences now. We have to be able to deal with these data to understand how the human body works. We have to understand how diseases work, and we need data to develop better and more effective treatments for these diseases. And with all these data, we have the opportunity to develop treatments for diseases also at the personalized level. For example, if I take a particular medication for a particular disease, it will work differently than if the person standing next to me takes that same medication for the same disease, because we have different genetic makeup, we live in different environment, and we will respond to these treatments differently. And so, but with all these data that we have the opportunity to leverage it to, for example, personalize our uh, treatments for various diseases. And so biology is no longer just biology and life sciences. This entire field now relies on the biological insights we learn working in the wet lab, that produces data that are then fed into technologies. These technologies are used for computation, generating new data, generating new hypotheses. Then these hypotheses are taken back to the wet lab, to the biological studies, and the cycle repeats. We generate new data, better data, more accurate data, that we feed it back to the technology computation, and the cycle goes on and on and on. And in this whole process, we end up generating new technologies because we need them, because we, for example, can't handle certain types of data or the amounts of data. So we develop new technologies, we develop new software for similar reasons, and so forth. And I'll talk about examples of this throughout my journey too, how we end up developing new software and new technologies. Um, and so going back to the original questions, why do we need computational biology? And so we need computational biology and computer models because biology is extremely complex. We have lots of data, but also not do we not only have lots of data, these data and pieces of information, these genes and proteins, they form these very complex networks that are very hard to understand just by looking at these pictures. And so we need computer models that allow us to understand, to simulate, for example, what happens if you change the environment, you add, more of something, you subtract something else. How does that impact these networks? If you were to, look at this picture right here, and you try to say what happens if you increase this here, this area here, how does that impact something else on this side? It's very difficult to do that by looking at these pictures. And so the computer models allow you to do that. They also allow you to look at what happens if you perturb something in these pathways, if you block something, how is that going to impact the rest of the system? How does providing a medication or drug for a disease, how does that impact the rest of the system? And using these computer models has the potential then to give us better insights, for example, into the side effects that potential treatments can have on a person or a population, because these treatments and drugs, they indeed affect these components of these very complex biological networks. And so with computer models, we can look at what happens to the rest of the cell, what happens to the rest of the organism. But because it's a computer model that can be run relatively fast and much faster than, for example, a clinical trial would be done, or much faster than a wet lab experiment can be done, or a mouse study can be done. We can run any kinds of combinations and crazy configurations on the computer model. We can run simulations and ask questions. What happens if you combine two drugs? What happens if you combine three drugs? What happens if you combine them in different sequence, different order, different dosages? How does all of that affect the human or the cell or these complex networks? And with computer simulations, it's relatively easy to do that. It's substantially faster to do that than in the wet lab. And so this is one of the primary reasons that computer models, or one of the very powerful utilities of computer models. And so going back to now my story, when I was an undergraduate student with Jim Rogers, and remember he hired me to help him build such a model of signal transduction. And so I did that. I didn't know at that time, I had no idea how to do it. I knew math and I knew some computer science, I knew some programming, but like I said earlier, I didn't know much biology and biochemistry. And so he asked me to build a model of signal transduction. And so here is the network diagram of that model. Each one of these labels, those are different proteins that signal to each other. These at the top are signals, we call them ligands, that stimulate this signaling network. And so this happens, this spaghetti code is inside a cell, and then these blue rectangles are outside of the cell that provide those signals and tell the cell to do something. And so when I started building this model, I had no idea what any of these things were. They were just labels to me. And Jim told me, well, go just start reading papers. And I said, where do I find these papers? And he said, go to PubMed. And I said, PubMed what? Computer science student. I didn't know where to find scientific literature. So I literally started at square one. Okay. And I started reading papers. I started finding papers, started reading papers. And I was going back and forth asking him, what is phosphorylation? What does this mean? What does that mean? And over the years, I was able to build this model. And I ended up reading about 600 papers to be able to put this model together. And this is a relatively small model. It has only or had 130 of these signaling components and about 500 interactions, these edges that indicate how these proteins communicate with each other. But I read lots and lots of papers that gave me the opportunity to really dive into biology. And so we built this model, and so I had to learn some additional math to be able to describe this complex biological mess mathematically. And I also had to, we put this model together, and then the idea was to simulate the model to see what happens under these different conditions. And but there were no ways, no software tools to simulate it, and so I had to learn how to program in C++, plus plus. And that was, to me, that was more of where I felt like at home because I was still a computer science major. And at that time I was learning Java, or I might have been learning C++ in a course, and so this gave me an opportunity to really to further develop my programming skills. And I also had to learn how to develop a database to hold all the knowledge about this. So when we build these models, and I'll talk about that a little bit later, it's extremely important to understand why these components are in here. Why are these connections in the model? We have to be able to annotate them so that when we share this model with other scientists, they know why we added this link in there. And we add the link in there because it was described and characterized in the scientific literature. And so we need to be able to track all this literature and all these pieces of evidence that we use to construct this model. And so I built the database to be able to do that. And so it was at this time that I started, I really started realizing how I can combine my passion for computer science and mathematics with biology. And it, in fact, gave me a new purpose, I suppose, because up until that point, I felt like I was going to graduate with a computer science degree, and I was going to be writing software in a bank, and it just didn't seem very interesting or fulfilling. And so, being part of this, having the opportunity in this research experience really opened my eyes to a field that I had no idea existed before. The ability to combine, computation and biology together just developed into a whole new passion for me. And so during my undergraduate degree, I switched from computer science to bioinformatics. I think I was the first class of the bioinformatics undergraduate program at UNO. And like I said, that whole experience and then that program really put me on a completely different trajectory. And I don't even look back on that. And so I finished my undergraduate degree in bioinformatics at UNO, and then my plan actually was to go work for a pharmaceutical company, go get an MBA, do an MBA first, and go work for a pharmaceutical company and make lots and lots of money. Well, Jim had a different idea for me. Jim once again influenced me in my life, significantly so, and told me, you have the commitment to do a PhD. And I had never considered doing a PhD. Uh, but after talking with him, and I was young, and I said, I don't know if I can spend another 5 years of school. I want to be done. I want to go, live my life. And but nonetheless, he convinced me to do a PhD. And actually, to this date, I think that's the best decision that somebody almost made for me. Because again, being able to dive into computational biology and really learn about science and learn about diseases and biology in general, uh, just fostered and developed this passion in me. And so I again, I don't even look back, and now I think it would have what a potential disaster it could have been had I gone in a different direction. And so in 2006, I joined the Department of Pathology and Microbiology at UNMC to do a PhD. And in that time, I continued to work with Jim. Jim then became my PhD advisor. And we continued to work on the model that I started as an undergraduate student, and I finally finished it. And we were able to use this model. And the reason why we were building the model to begin with is to understand how cells process information, and how these signaling networks is what provides this decision making and information processing ability to cells and biological systems. And so in 2008, I was fortunate enough to publish my very first paper. And that really has seeing your name on your very first publication is is just it's an undescribable moment again, and one that really shows you um, whether you want to go through all this drudgery work to accomplish this. And I now tell my students, research is about 90% drudgery work and 10% reward. When you see your results come together and your name on that publication, and you have to be committed, you have to be doing it for that 10% of that reward. And so that really that feeling when we published this paper really reinforced in me that this has been the right path for me, and the one that I want to continue going. And so when I finished my PhD, I went to the Department of Mathematics at UNO, and at that time I started developing more models. I started applying computational modeling to cancer, to looking at signal transduction in cancer cells. And we published a paper with a couple of researchers at UNMC, cancer researchers. We also developed an influenza model. What happens when influenza infects airway epithelial cell, how that hijacks the signaling pathways inside the host, and then uses those signaling pathways to replicate itself and propagate the infection. We modeled HIV infection of other cell types, macrophage cells. And it was at that time that I started really pushing the boundaries of, how big of models can we really build. And so this was a model that this is a static picture of the network representing the model of signal transduction in macrophage cells. This was in an effort, as I mentioned earlier, to build a HIV macrophage host pathogen interaction model. And so we started building this model, And this student, Alyssa, helped me build this model. And started realizing, okay, this model is getting so big that we don't have really a good way to now visualize it. We can't just print it out on a normal piece of paper and look at it. If we want to trace those lines, it's very difficult to do. So we try to print it out in some readable way, and this is what we got. This is it took up about half of our conference room in the mathematics department. I had to roll it out like that to be able to follow these lines. And so that's when I realized really that this is when we get to this point, when we have so much data, so much biology, we can use the existing technology for so long, but then we continue to push the boundaries, and we get to a point where we need to develop new technologies and new software. And so at that time, we realized that it's not feasible to continue to build these models one at a time, or one person working on it at a time, because we spent so much time to build that model that we couldn't even print out on a piece of paper. We had to print it out on stacks of papers and then glue them together to even visualize it. And we realized that there's really, and even that model of that size, that was at that time, that was the largest model ever created, largest mathematical model that describes the mechanism of what's going on inside a cell that was ever created. And yet it was still just a little tiny fraction of the cell. And so we had the idea of, why don't we create software where experts working in these different parts of the cell and parts of the disease and parts of the body can work together to build these models with in some kind of software, in an open source, in a collaborative and open source fashion. And hopefully one day, then, if we have this collaborative effort, we can build a complete virtual cell, right? And so that's what I started at, during my postdoctoral studies, and then the idea was born. We had some programming, we had done some work, but then it was my time to move on. And so in 2013, I joined the Department of Biochemistry at the University of Nebraska in Lincoln. So at this point, I have been on 3 campuses of the University of Nebraska system, and I've I've continued to learn new things in every one of these campuses. And when I came to UNL, I continued to develop Cell Collective. And so Cell Collective is that software that we've created to make modeling more collaborative. And so when we built Cell Collective, the first thing that had to really happen is that, as I was learning how to model these biological systems, I did it by writing the mathematical equations and writing C++ code and so forth. But it's become clear that if I want a cancer expert to be able to start building or contribute to our models of signal transduction in cancer, they will they don't have the training in modeling, and they don't have the training in computer science, and it's not feasible to expect them to go learn how to program. We have to make modeling more accessible to anybody with any background. I've realized throughout my career that I feel that computational biology and bioinformatics have been developed by computational biologists for other computational biologists. And I feel like that has been a mistake. I feel like that is a limitation to being able to fully leverage the power of computational biology. I feel like computational biology is like you having to be a car mechanic to be able to drive a car, right? You don't have to be a car mechanic to drive a car. You need to understand the assumptions associated with driving a vehicle. You need to understand that if you press the brake pedal, the car will slow down and eventually stop. If you press the gas pedal, it'll start going and go, faster and faster. And I feel like computational biology and computational modeling need to be that way as well. They need to be tools that are accessible to anybody with any background, assuming that that person or the user understand the assumptions behind these tools. And so I've built Cell Collective for that purpose. That was the first founding principle of Cell Collective, is to make modeling attainable to anybody. And then collaboration and scalability. Now, if the modeling is accessible to anybody, to biologists who don't generally have training in computer programming and differential equations, then we can create an environment for collaboration, and we can scale these models. We can make them not only bigger but also more accurate, because now looking back, as an undergraduate student, I was reading hundreds and hundreds of papers, but I was not an expert on, you know, growth, EGF receptor signaling pathways, or other types of pathways. I was putting into the model what I was reading from these papers, right? But I'm not an expert on any of these pathways. And there is so much information in the heads and the minds of these scientists who spend decades and their lifetimes of working on these pathways. And so if we can get the experts working on on the individual parts of the biological system or organism, we can get even more accurate models. And we also want to make these models transparent. Uh, as I mentioned before, we want to be able to annotate why these different components are in there, and why the interactions are put in this way or that way. Where did that come from? And then that, by having transparent models, we can enable reusability and reproducibility of these biological systems and of these computational models. And so these are four principles that Cell Collective, that we've been really paying attention to when we design any feature in Cell Collective, and when we continue to develop this software. And it is in fact the case that these are extremely important ideas and concepts in the field of computational biology, that some scientists have their own research projects related regarding the reusability and reproducibility of data and computational models and bioinformatics software tools and the pipelines. Just like in wet lab research, we develop protocols so that anybody, when we publish a paper with a wet lab experiment and the results associated with that experiment, there is a protocol that we can usually share with others so that they can more easily reproduce these results. In computational biology, there are groups who are developing standards and standard protocols so that anybody can reproduce these results more easily. And so Cell Collective, so I'll talk to you about now Cell Collective, and just to give you an idea of, I've been interested in the biology and the biological systems and how they behave, and uh, how we can address different diseases. We've worked with, HIV, influenza, cancer. Now we work a lot in immunology, and I'll talk about that a little bit in just a bit. But before we could, before I could pursue these systems at a larger scale, we had to develop new software, right? And so this is Cell Collective, and also I'll walk you through some of these features just quickly. We developed Cell Collective initially to make it to make modeling attainable to anybody, and focusing on researchers, on scientists, to help us build these models. But in the meantime, we also have realized that our students, by having our students build models just like I did personally when I was that undergraduate computer science student, that's how I really learned uh, biochemistry and biology. This was during my undergraduate research, as I mentioned earlier. I read, hundreds and hundreds of biochemistry paper and biology papers. And then when I was taking my PhD classes, uh, BRTP 824, I still remember to this day, it was on signal transduction. And that was probably the easiest class I have ever taken, because I learned most of that stuff before just by building the model. And so I realized that you can learn a lot of biology by building computational models, and by making modeling attainable to anybody. Now suddenly students in biology classes can learn biology, and they can learn or appreciate the dynamical nature of biology and its complexity, the network complexity of biology, by building and simulating and breaking computational models too. So users can easily just build models by adding these components, and these components again can be signaling components, or they can be genes, or they can be persons, they can be cells, they can be organs, depending on what it is that you're modeling. And so you add these components, these dots, and then you start connecting them with edges. You literally draw an edge from one component to another component, and you don't see. Now I have this panel here that shows you the mathematics, but the default behavior, you don't see any math, but yet as you're doing all of this, we're building a mathematical model in the background. And you don't have to see the math if you don't want to, but you can if you're interested. If you're a computational biologist or a computer scientist, you can see the math and you can interact with the mathematics in the background. And then we have a simulation, and so you can now. So we have this particular model is a food web model. So uh, we've got coyotes who eat rabbits, um, rabbits that eat seeds, and so forth. So this is an ecosystem. This is a level at the ecological uh, layer. And so then you've got the simulation. These are sort of activities associated with each of these components, and then we've got some environment here that we can change the parameters of. We can, we just introduced the deer disease, some kind of a disease that kills deer, right? And so we can see how that starts impacting. So you'll see that the deer population goes down, but suddenly you have more flowers because there are less and less deer to eat flowers, right? And so you can start looking at the impact of changes in the environment or perturbations in the system on the dynamics of the remaining parts of the system. And so since we've developed Cell Collective, it contains, this is a little bit of an outdated figure, but it contains now lots and lots of different computational models, that models of biological processes, of signal transduction, gene regulation, metabolism, ecological systems, and so forth in different species and different cell types. We've got T cells, we've got different pathogens, plants and various organisms. And so users can now, anybody, biologist or computational biologist, anybody can really start building models, or they can use these published models that the scientific community has developed and published, and they can start expanding on them, and they can start questioning them, their accuracy and their correctness and so forth, and they can start improving on them. And so today, Cell Collective is used by hundreds of scientists all over the world, and as I mentioned before, also in education. And so we now have Cell Collective has been used in over 70 institutions where they use it for purely education purposes. To teach, where students learn about gene regulation and cell differentiation and glucose homeostasis and many other processes through in biology and biochemistry classes through these modeling and simulations. And so again, our goal is to make modeling attainable to anybody. It doesn't matter if you're a scientist, it doesn't matter if you're a student, it doesn't matter if you're interested in computer science or bioinformatics. If you're interested in understanding biology as a system, as a dynamic process, I want you to be able to use modeling. And in the classroom, we want to be able to teach students with the tools that we use as scientists and as professionals, as experts. Once we graduate with our degrees, we want students to be able to practice and learn through a constructivist and inquiry-based and scientifically authentic manner. Okay, instead of this. And now thinking back, I mentioned earlier that when I was an undergraduate student, biology was not on the top of my favorite classes, and I now retroactively realize why, and that is because to me mathematics and computer science were more exciting because I got to do things, I got to build things, I got to break things and mess up things. But unfortunately with other classes, it was more about reading textbooks and being and listening to lectures. And so we feel like now with computational modeling we can also provide a different way that students can engage with biology, and they can appreciate the dynamic and complex nature of it while doing so. And so going back to, so we've been going in the circle throughout my career over and over again, and then we realized, we need to develop new software and new technologies, and now that we have Cell Collective and a lot of other tools we have developed in the course, Cell Collective was sort of an example of how we go around the circle and how we realize, oh, we need a better software, we need a better technology, let's build it, and let's go back to the biology, let's go back to asking biological questions, and let's go back to understanding human diseases and biological processes better. And so over the last couple of years, I have become very interested in immunology, and our group has really started focusing on the human immune system. And to me, the human system is extremely fascinating. It has to be precise. It is complex, but it has to be precise. It has to maintain homeostasis in a body. It can't be overreactive. It can't be lacking in the reactivity when we get infected, or when a foreign object gets into our body. the immune system has to be able to fight it, but it can't fight it too much, because then if it fights it too much, then we have a autoimmune disease. So the immune system starts attacking our own body. We don't want that either. So there has to be that right balance. And to me, that the maintenance of this healthy balance is very fascinating. And it's fascinating also because of how it's done, how that balance is achieved. So the immune system is composed of a number of different immune cell types, and these immune cell types, these immune cells, they talk to each other, they communicate with each other through molecules such as cytokines and different chemokines. They become, these molecules become the communication channels between these different cell types, and then they mediate that immune response to a foreign object entering the organism or our body. And so I want to be able to understand how all of this comes together to give rise to a proper immune response. Well, if you can probably by now guess where this is going, but yes, the immune system is also a very complex network. All of these different cells that I mentioned earlier, and these different cytokines and interleukins and so forth, they form this complex network. And so, again, if you want to understand how, what will happen if you want to if you want to downregulate, if you have a medication, for example, if you have a medication to suppress the immune system for a transplant, so that the immune system doesn't attack the transplanted organ, you have to suppress the immune system. But the immune system is suppressed in that particular case by suppressing CD4 positive T cells. And but you can see these CD4 positive T cells right here. They're connected to all these other different cell types, and so you start throwing the immune system and the homeostasis out of whack, because you're messing with one part of it. And so again, to understand how that suppression of the immune system here can affect something else that may or may not be predictable, we need a computational model of that. And so, with the immune system, it is a multi-scale problem, because the immune system at the level of cells, and as they communicate with each other, they form a complex network. We have in each of the cells, we have genes that create complex gene regulatory networks. We have, I've talked about signaling networks at the protein level. We've got complex, signal transduction networks. The different organs are also impacted by the immune system, and that those also form a network. And also where we live in our environment, and who you can probably relate now during the coronavirus situation that we have, crisis that we have right now, is that it matters who you interact with, whether you can help suppress the spread of the infection. And so again, it's all multi-scale. It's the immune system and how it protects us or doesn't protect us. It happens at many layers of biological organization. And so trying to understand the immune system has been one of our goals, and we can't do it all at once, and so we're focusing on one layer at a time. As I show you, this is sort of at the cellular level what happens, how the cells communicate with each other. But when we start now, my postdocs and my students, suffer with me maybe, because now they are the ones reading hundreds of papers to build these models. And uh, and when they started reading these papers, they realized that immune system is not as simple as this diagram, but it's more complex, something like this, right? And so we've been able to build this model that contains 26 different types of immune cells, and 35 different cytokines and other components of the immune system, and how they all interact with each other. And we build this model now to be able to study different types of infections, to see how they stimulate the immune system in different ways. We can start looking at how two infections, for example, together elicit different or similar, that's a question, immune response than if you just have one at a time. Does the order of infection matter to the immune system or the immune response? And so we can be asking questions like that. And we can look at, we can, the similar heat maps you will see in a wet lab experiment now with high throughput data. But we have our own computational model that can produce lots and lots of data under an infinite number of perturbations and combinations of different parameters, whether it's the pathogens, whether it's drugs, or random mutations, and so forth. And so we can start looking at how does the Ebola virus, what does the immune response look like when you have Ebola infection? What does it look like when you have MTB infection, and so forth? And so I am running out of time here, and so our long-term goal is a crazy goal, but our long-term goal, and I truly believe that it can be done, is that one day we will have a simulatable human, one that can be used by scientists to continue to learn and understand the mechanism of diseases, the the foundation, the basic principles of how our cells work, how the immune system works, how everything inside of our body works, but a model that can also be used by clinicians and doctors to put all these data together to be able to come up with more accurate diagnoses, or to be able to adjust and personalize drug treatments for, at the individual or subpopulation level. And but as I mentioned, hopefully by now it's clear that the complexity of these biological systems and diseases, and the amount of data that goes into the biological complexity, is why we need computational biology and bioinformatics. And really, it's the combination in life sciences. I believe that I'm convinced that the next generation of health professionals and life science experts have to really understand the intersection of biology and computation, and computer science and mathematics, because of where we are with the data and the important role that data and computation play in uh, in today's and tomorrow's life and health sciences professions. And so with this, I would like to end. Thank you very much for your time. And before I do, I want to point out that all of the work that I've discussed, and the journey, has been really the work and a result of the impact and influence of people around me, whether it was Jim at the very beginning of my career, or the different positions where people hiring me took the chance on me, and most, and also the members of my team, the different postdocs and software developers and graduate students and undergraduate students, all of those, and of course the funding agencies that enabled us to do all this work. It's really a product of all of these different things and different people coming together. And so I would like to end with this. Thank you very much for listening to this presentation. And if you have any questions at any time, please don't hesitate to reach out. You can use this email address and get in touch with me. Thank you so much. Bye.